In the last class, we studied about the exceptions for the law of dominance. In that, we studied about incomplete dominance and codominance. When we studied codominance, I also explained you about the concept of multiple allelism, where in case of human blood group, the expression of the blood group is controlled by not only two alternatives, there are three alternatives, that is IA, IB and small I. Now let us switch over to another concept which Mendel did not observe. According to Mendel, there is a unit factor which controls the expression of only one phenotype. But later on, it was found out that sometimes one gene can control the expression of more than one phenotype. That is called as pleiotropy. Let's see. In case of the pea plant, the seed size and seed shape, they are controlled by the same gene. And both the phenomena, they don't show the similar law of dominance. One feature shows complete dominance and other feature shows incomplete dominance. Let's see through the cross. Here, let's first take seed shape. When it is capital B, then the seeds are round. When it's small b, the seeds are wrinkled. And when they are crossed, in F1 generation, all the seeds are round. That is complete dominance. And when these round seeds are crossed, in F2 generation, we get this same ratio 3:1. 3 parts are round and 1 part is wrinkled. So this is law of dominance. But the same gene controls another character also, that is seed size. When it is capital B, the seeds are large in size. When it is small b, the seeds are small. But in F1 generation, all the seeds are intermediate. This is incomplete dominance. And when these intermediates, that is F1 generation, are selfed, then in F2 generation, the phenotypic ratio is 1:2:1. That is 1 part large, 2 parts intermediate and 1 part small. So this is incomplete dominance. How does this take place? Let's find out. Now let us study the actual reason behind these two different types of phenotypic expression. Actually, both these phenotypic expressions, that is seed shape and seed size, is due to a single gene product, which is starch grain. When it is capital B, then starch grains produced are large. As a result, the size of the seed is very large and its shape becomes round. In contrast, when it is small b, less starch grains are produced and they are very small. As a result, the size of the seed becomes very small and its shape becomes wrinkled. But in third case, that is capital B, in heterozygous state, intermediate size of starch grains are produced. As a result, the size of the seed is also intermediate and its shape is round. So, which appears to be two different phenotypes, actually are the product of one gene product. So, we can say that dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on particular phenotype that we choose to examine. Okay, so this concept is very important. Now this concept of pleiotropism is also given in your book with respect to one disease, that is phenylketonuria. This phenylketonuria is caused due to the gene mutation, a single gene mutation, which is responsible for the production of the enzyme phenylalanine hydroxylase. This single gene mutation also affects the manifestation of other phenotypic expressions like mental retardation and reduction in hair and skin pigmentation. Therefore, it is also a type of pleiotropic expression. So, after pleiotropy, let us understand about this concept, explanation of the concept of dominance. How this dominance is able to express itself. According to Mendel, he had given the paired alleles concept. He said that for every trait there is a paired alleles, and these paired alleles may be homozygous dominant, homozygous recessive, or heterozygous. Later on it was found out that each allele is actually a gene. If that gene is normal, or you can say dominant, it will be coding for a normal enzyme, and that normal enzyme will be responsible for physiological reactions, and the expression of that phenotype will be there. So let us see what all can be the cases. There can be 4 cases. First, as I mentioned you, one allele is normal, other allele is also normal because it is producing a normal enzyme. So the phenotypic expression will be dominant. But sometimes there can be the production of a less efficient enzyme. In that case, the expression will be intermediate. Some work will be there, some phenotypic expression will be there, but it will not be complete. And the phenotypic expression will be called as intermediate, as in case of incomplete dominance. Then there can be a non-functional enzyme. Enzyme is not produced at all. So in that case, it will be recessive. Also, if no enzyme is produced, here function enzyme is not there, here no enzyme is produced. In both the cases, it will be recessive. There will be no expression of that particular trait. So these are the reasons for the concept of dominance.